Alexa, Isaías, and Yair from Team 7 of Group 4 of Bacteriology class presents Detection of Clostridioides difficile through Test-PCR-LAMP. What's the deal with Clostridioides difficile? At the microscopic level, Clostridioides difficile is a rose-shaped bacterium, slightly curved or straight. Clostridioides difficile is around 1 to 1.5 micrometers in width and 3 to 8 micrometers in length. Clostridioides difficile belongs to the gram-positive bacteria. One distinctive characteristic of Clostridioides difficile at the microscopic is its oval-shaped endospores that are near the center of the cell. Clostridioides difficile does not possess flagella or other movement structures. Clostridioides difficile is a pathogen capable of generating an infection called CDI, Clostridioides difficile associated infection. From now on, we are referring to this infection as CDI. The CDI susceptibility is due to a wide range of factors, such as the recent antibiotic use, advanced age, underlying medical conditions, or immunocompromised status. Disruption of the normal gut microbiota, often due to antibiotic therapy, creates an environment where Clostridioides difficile can proliferate and cause infection.
The CDEs follow the next natural history. In prepathogenesis, Clostridioides difficile is transmitted via fecal-oral. Its spores are ingested via contaminated food, water, or healthcare environments. In pathogenesis, once ingested, Clostridioides difficile spores germinate into vegetative cells in the colon under conditions like dysbiosis. The cells produce primarily toxins A and B. Clinical disease manifests when the toxin-producing Clostridioides difficile reaches a critical threshold in the gut, resulting in symptomatic CDE. The severity of symptoms can vary from mild diarrhea, abdominal pain, pseudomembranous colitis, toxic megacolon, or life-threatening complications, depending on factors such as the strain of Clostridium difficile involved, the patient's underlying health status, and the presence of predisposing factors. During the diagnostic phase, healthcare providers utilize diagnostic techniques such as PCR-LAMP to detect the presence of Clostridioides difficile, DNA, or its toxins in stool samples from symptomatic patients. Then, in resolution, disability, or death, with timely diagnosis and treatment, many cases of CDE resolve without complications. However, severe or recurrent CDE can lead to prolonged illness, increased healthcare costs, and even mortality, particularly in vulnerable populations.
And why is Clostridioides difficile so infectious? These bacteria possess a long list of virulence factors, such like toxins A and B. Toxins A and B are the primary virulence factors produced by Clostridioides difficile. These large exotoxins are responsible for the characteristic symptoms of CDI. They function by disrupting the cytoskeleton of intestinal cells, leading to cell death and mucosal damage. But Clostridioides difficile produces adhesins that facilitate its adherence to intestinal epithelial cells. Spores formation. Clostridioides difficile has the ability to form spores as a survival mechanism, resistant to environmental stressors such as heat, desiccation, and disinfectants. These spores persist in the environment for extended periods, awaiting favorable conditions to germinate into active bacterial cells and grow. They can persist in the colon and cause reinfections. Regulatory factors. Clostridioides difficile produces regulatory proteins that control the expressions of virulence factors and other pathogenesis genes, which helps to adapt to different environmental conditions.
How do we know if there's a CDE? Clostridioides difficile possesses conserved in specific regions like ribosomal RNA encoding, necessary for the bacterium's growth, metabolism, replication, and pathogenesis, including colonization, production of toxins, and immune evasion. Techniques like PCR-LAMP assay can target those conserved regions. PCR-LAMP is a molecular diagnostic technique that amplifies specific DNA sequences even at low concentrations, and is relatively simple and fast compared to traditional PCR methods. Once the target DNA sequences are amplified, they can be detected with visual inspection of turbidity or fluorescence-based detection systems. Genes encoding for toxins A, TCD-A, and B, TCD-B, are specific to the Clostridioides difficile genome. Those toxins are produced as precursor proteins of active toxins. Toxins A and B exert their cytotoxic effects by targeting and modifying specific proteins within intestinal epithelial cells. For example, TCD-A and TCD-B are glucosyl transferases that inactivate small GTPases, leading to cytoskeletal disruption, cell rounding, and cell death. These toxins disrupt the integrity of the intestinal epithelium, causing inflammation and inducing CDE's characteristic symptoms. The PCR-LAMP test typically utilizes a reaction mixture of various components, like a DNA template extracted for clinical samples, primers targeting specific regions of interest within the DNA template, DNA polymerase enzyme with strand displacement activity like best DNA polymerase, deoxynucleotide triphosphates DNTPs for DNA synthesis, buffer solution to maintain optimal reaction conditions like pH and cell concentration, and fluorescent dyes like ACE, YBR green for real-time monitoring of amplification.
Clinical samples to be used, such as stool specimens, are commonly used for the detection of Clostridioides difficile DNA or its toxins. Samples should be collected using appropriate collection devices and transported to the laboratory under recommended conditions to preserve DNA integrity. Upon receiving the laboratory, samples may be processed to extract DNA using commercial DNA extraction kits or other methods. Extracted DNA is then used as the template for the PCR-LAMP assay.
The PCR-LAMP test follows the next process. Initiation. Outer primers F3 and B3 bind to the target DNA sequence, initiating DNA synthesis. Then self-priming DNA synthesis. Inner primers FIP and BIP bind to the newly synthesized DNA strand, leading to self-prime DNA synthesis. Then DNA polymerase extends DNA strands by strand displacement, displacing inner primers. Then amplification proceeds exponentially, generating large amounts of DNA. Loop primers, loop F and loop B, accelerate amplification by binding to the loop structures in the DNA. Finally, amplification is monitored in real time using fluorescent dyes like SYPR green that bind to double-stranded DNA. The entire process occurs externally at a constant temperature, typically around 60 to 65 degrees Celsius.
The technique uses different experimental controls. Positive control contains known DNA template, positive for the target sequence, ensuring the proper functioning of the assay. Negative control contains no DNA template, serving to monitor for contamination or false positive results. Test performance control monitors various aspects of the assay performance, such as reaction efficiency and inhibition. Resulting samples consider positive, negative, or doubtful. Positive result: presence of side-modal increase in fluorescence, real-time monitoring, or leather-like pattern on gel electrophoresis indicates amplification of the target DNA sequence, indicating a positive result. Negative result: no amplification or minimal amplification indicates the absence of the target DNA sequence, indicated a negative result. Doubtful result: ambiguous or inconclusive results might require further investigation or retesting to confirm.
PCR-LAMP assays for Clostridioides difficile detection are highly sensitive and can detect even low concentrations of this bacteria DNA in clinical samples. Sensitivity levels typically exceed 90%, meaning that the test correctly identifies a high proportion of true positive cases of CDE. These tests also are highly specific, often exceeding 95% values, meaning that the test has a low rate of false positives for both screening and confirmatory purposes. In screening scenarios, PCR-LAMP can rapidly detect the presence of Clostridioides difficile DNA in clinical samples, helping to identify individuals at the risk of infection or colonization. In confirmatory scenarios, PCR-LAMP can be used to confirm the presence of Clostridioides difficile DNA in samples that have tested positive with other screening methods, such as enzyme immunoassays for Clostridioides difficile toxins. Some advantages of the test are rapid detections, often gives results within 30 to 60 minutes compared to traditional culture methods, which can take days. Also have high sensitivity and specificity, allowing for accurate detection of Clostridioides difficile even at low concentrations and minimizing false positive or false negative results. It also has a simple protocol and can be performed using basic laboratory equipment, making it accessible to a wide range of laboratories. It's an isothermal amplification that eliminates the need for complex thermal cycling equipment, reducing the cost and complexity of the assay setup. Finally, it has a real-time monitoring of amplification, enabling researchers to track the progress of the reaction and analyze results more quickly.
Disadvantages of the test: risk of contamination, which can lead to false positive results. Proper laboratory practices and contamination controls are essential to mitigate this risk. Potential cross-reactions: although PCR-LAMP assays are designed to be highly specific, there is a risk of cross-reactions with closely related organisms or non-specific amplification, particularly if the assay target conserved regions of the genome. Handling requirements: proper handling of clinical samples and reagents is critical to ensure the accuracy and reliability of PCR-LAMP results. Inadequate sample preparation or storage can lead to inaccurate results. Several commercial suppliers offer PCR-LAMP kits for the detection of Clostridioides difficile, including Aiken Chemical Co. Ltd. Aiken offers the LUPAM DNA amplification kit, which includes reagents for PCR-LAMP assays, targeting various pathogens, including Clostridioides difficile. Optigen Limited. Optigen offers the Gen 3 real-time PCR instrument and associated kits for isothermal amplification, including the detection of Clostridioides difficile. The cost per test for PCR-LAMP kits vary from a few dollars to $10, depending on factors such as the supplier, kit configuration, and volume discounts. Thanks for watching.